everyone. In this chapter, we will continue with our survey of the phyla and groups in the animal kingdom. Last chapter, we talked about the vast diversity of invertebrates we have. This chapter, we will focus on the origin and evolution of all the vertebrates. Now, before we start, I wanted to make a note that I have skipped over the last section of the chapter that talks about all the different fossils of humans, the Neanderthals, and all the specific scientific names of all the hominins. Hominins are, it defines as the fossils of human Homo sapiens. So it's a pretty interesting topic, but it is very long. So I have to let that section go due to the length of the chapter. But know that you are always welcome to read all about it from the textbook. So let's get started. All right, so one of the heaviest animals to ever walk on land was the plant-eating dinosaurs as massive as 40,000 kilograms, or they say it's equivalent to 13 pickup trucks. So you can see why vertebrates, which defines as animals with a backbone, is one of the most successful groups of animals on Earth. Um, there are actually only, though, 60,000 species, but there are enormous diversity in the traits that animals have. Like I've said, we have, humongous animals with very different body mass. The biggest animal to ever exist on Earth is also a vertebrate, known as the blue whale. And they weigh more than 100,000 kilograms, which is really, really even more heavy or heavier than the plant-eating dinosaur. So at the same time, there are also fishes that are 100 billion times smaller than a blue whale. So with that example, you can see how the huge amount of diversity we can have with vertebrates alone. So just because vertebrates have less species, it doesn't mean they are less interesting to study. So what are some key traits that appeared during vertebrate evolution? Well, first of all, there were skull, and vertebrate backbone that are found in all vertebrates. So that is something new when we compare to invertebrates. Back when we talked about in the last chapter, all the invertebrates did not have a skull of any sort, neither do they have a backbone. That's why they're called invertebrates. Second thing is that there are jaws now. Jaws have started to appear with vertebrates, and also mineralized skeleton. Mineralized means that there's calcium within the bones. That's also something new because all along we either don't have bones at all, or we have bones that are just made up of cartilage. So and we will have some of that still in the early groups of the vertebrates. But once, we get to the higher, more complex vertebrates, then we get these really strong bones due to deposits of calcium. And it is these two things, having jaws and mineralized skeleton, that enable vertebrates to become one of the most dominant predators of all species on Earth. Third, there are an appearance of lobed fins. What are lobed fins? They are muscular fins with rod-shaped bones. Now, the rod-shaped bones is something that is special in these group of species, and they are specifically called lobe fins in the groups that we'll talk about, such as lungfishes. They have these masculine and really big fins that are, sort of for the purpose of, being stronger and being able to even dig in mud. We'll talk about them when we get there. Fourth trait is that there are limbs with digits. Digits is another scientific name for having fingers. Just remember that because the book and continuously refer to fingers as digits. So when we're talking about digits here, we're not talking about numbers. We're talking about fingers. So there's limbs now with fingers, and also if we're talking about our feet, there are toes. So those have evolved from lobe fins as well as a very, very important and critical development for animals to colonize the land. So we'll talk about how that's the case when we get to that specific animal phyla. And last but not least, we have amniotic egg. Okay, so there are group of animals called amniotes that we'll talk about. And that is part of the amniotic egg that can protect their embryos, and then again enabling the vertebrates to have some sort of reproduction on land. Without those amniotes or amniotic eggs will dry out because it is prone to dehydration when it's just a wet surrounding, because a lot of the embryos we have in species in animal kingdom especially, they have to be encased within some kind of fluid for diffusion of things, and also to make sure that, before our skin is developed, furs develop, all that stuff, the embryo is just tissues, and those tissues are prone to drying out if they don't have any sort of protection, which is why we need an amniotic egg of some sort. So we'll talk about that feature when we get to that particular group of animals. All right. So vertebrates are part of the phylum called Chordata. We have talked about that from the last chapter, and we refer to these as commonly as chordates. All animal species that reside in the phylum Chordata, you can say they are chordates. And all chordates have bilateral symmetry, or you can call them bilaterians, and they fall under the group of deuteriostomia, which is why we didn't talk about much about chordata, the phylum chordata, in the last chapter, due to the fact that we'll talk talk a lot about them here. So there are two groups of invertebrate deuteriostomes that we didn't talk about in the last chapter, and I said we'll talk about it in this chapter. So here we are, because we have to talk about the things that they exhibit that lead to the features that vertebrates have. So that's what we have to talk about, those two invertebrate groups. On top of that, they say that these two particular group species, are more related to vertebrates than other invertebrates, which is why we also place them in this particular chapter. So these two group of species are called cephalochordates, or also known as landslets, and then the other one is urochordates, and otherwise known as tunicates. So this is the phylogenetic tree that we will keep referring back to as we go through the chapter. It also shows that these two species right here, Cephalochordata and Urochordata, are the closest relatives of all the animal vertebrate species that we'll talk about for the rest of this chapter. So we will discuss these vertebrate animals after lancelet and tunicates, in the order as listed in this particular phylogenetic tree. So there are four key traits shared between chordates. We're talking about generally amongst all chordates, which is why chordates is right here, that encompasses everything. So for the first one of those key traits is called notochord, which is longitudinal and a flexible cord or rod between your digestive tube and the nerve cord itself. I'll talk about what the nerve cord is in the next in the next section. And then it is composed of fluid-filled cells that are encased in the fibrous tissue. Also note that notochords are developed or formed from mesoderm, because we talked about the three germ layers in the previous chapter. So there's ectoderm, mesoderm, and endoderm. So notochords are actually formed by your mesoderm. So if we look at the example of lancelet, because this creature right here, odd looking creature right here, is actually the lancelet. We'll talk about them more detailed in a bit. But they use lancelet as the picture to point out the four key traits that is shared between chordates, because lancelet pretty much have these traits throughout its lifetime. So here's that notochord right here. Okay, this kind of sits dorsally, or more on the dorsal side of the animal, and then it is kind of squished between the digestive tract, or digestive tube in this case, and also the nerve cord. All right, so what is notochord's purpose for then? Well, it provides skeletal support for the length of the chordate, the entire chordate. Now, there are also more complex jointed skeleton that can develop around the ancestral notochord in most vertebrates, which is why notochord in these simpler species stay as notochord, but as we get to the more complex vertebrate species, then you will see that this nodal cord start to branch out and slowly develop into more complex looking things that we'll talk about later. Or sometimes in some species, the nodal cords is reduced or completely gone. So we'll distinguish that when we get to those species. All right. Okay, so like I said, for specifically in humans, we have actually reduced nodal cords. And these reduced nodal cords will form something. It forms actually the central parts of what we call the intervertebral disc. And these intervertebral discs are usually cushioning between vertebrate bones. So here's a vertebrate bone. Think of it as looking at from the sideways. So this is the side of your back, or what we in anatomy and physiology calls it the lateral side view. So lateral view. So this is lateral view of the backbone. This whole thing here is one vertebrate bone. This is the second vertebrate bone. So what's sandwiched between there is then your intervertebral disc. So if you look at the vertebrate bone and the intervertebral disc from superior view or the top view, then you will see that there is this intervertebral disc, and that little cushion of intervertebral disc actually have two types of tissue. And I'm not going to talk about the other one here, but in AMP1, we did talk about these two, but the central core of that intervertebral disc is what we call a gelatinous mass of some sort to cushion, and that is called nucleus pulposus. So they say that nodal cords in humans have, sort of been reduced to forming just the nucleus pulposus of the intervertebral disc. And intervertebral discs actually serve as a very important key feature of keeping our back, these bones from, crushing each other, and that can be a big problem. And also there's a lot of nerves coming out of our back, so it's keeping everything in place and not painful when we move. All right, second key trait that is shared between chordates is what's called a dorsal hollow nerve cord. Now, remember that nodal cord and nerve cord are two different things. Don't get confused between those two. Now, nodal cord for generally in chordates are formed by a plate of ectoderm. So here's a picture of our, sort of be kind of between blastula, early blastula stage, beginning to go into, or late blastula stage, going into gastrula. So you have the blue layer here as the ectoderm. The pink layer is then your mesoderm, and then the yellow layer is the endoderm. So the outer layer right here, that ectoderm, would actually start to kind of pinch upwards, and then they will roll into a neural tube that becomes a hollow tube, and we call that a nerve cord that is located dorsal to the nodal cord. So if you look back at this particular picture of the lancelet, here's that nodal cord. Dorsal to that, or in back of that, or posterior to that for humans, in that sense, is then the dorsal hollow nerve cord right there, the yellow one. So this is just showing you how it's formed by these neural plates of your ectoderm. The cells start to migrate inwards into the interior part of the embryo, forming this nice indentation. And that indentation will then eventually roll up and complete, they kind of close up here, and then they will kind of pinch off and become your neural tube, neural tube or nerve cord in the, inside the embryo. So the nerve cord of the embryo, what would that become? Eventually, it should become the central nervous system, which in our case, we're talking about brain and the spinal cord. Okay, so central nervous system includes your brain and the spinal cord. So that's why the nerve cord, this formation right here is very, very, very critical process of embryo development, because if anything goes wrong with your central nervous system is going to be messed up. And when you don't have a proper correct position or even formation of the central nervous system, the embryo might not even survive. So it is very critical, this particular step, which is why they say, the first trimester of embryology, according to humans, is very, very critical because everything that is crucial for the survival of the baby is forming during that first trimester, including this hollow nerve cord. All right, third key traits of that share between chordates are called pharyngeal clefts. What are these? So pharyngeal clefts are grooves that separate series of what's called pharyngeal arches along the outer surface of the pharynx within the embryo. Okay, when I was looking at that on the text, I was like, what is that? Well, because we didn't talk about that, right? So I found a picture of the embryo, and you see these bumps right here. Okay, those bumps that you see are not fingers, okay? Because the hand is actually, the rudimentary hand is actually right here, that little nub. So these little nubs right here are actually the pharyngeal arches. And then the groove between these pharyngeal arches are then your pharyngeal clefts. Okay. And they say that these pharyngeal clefts will eventually become or develop into pharyngeal slits. And that is very critical when we're talking about formation of the gills. We'll talk about that when we get to the marine animals. But pharyngeal slits will is what allows water to come in from the mouth and then go through the body, exit the body, go through the body and exit out of it without ever passing through the entire digestive tract. Because think about, if you are a fish, you're constantly in water, but you still gotta breathe in water. So every time you open your mouth to breathe, water is just gonna come rushing through. If all of that water goes into your digestive tract, then they probably have to pee all the time, and a lot of things are going to go wrong in terms of osmosis. So this is why they have to skip over the digestive tract when they're just breathing and not necessarily drinking the water in terms of humans, if we're thinking about that. But so if you think about a marine fish animal, then they will need to have some way of bypassing the digestive system and only use the water for gas exchange purposes, respiration purposes. And that's what those pharyngeal slits are for. So if you look at this land slit right here, there's those pharyngeal slits because land slits do live in water. So there are aquatic animals, very simple aquatic animals that look like a little worm, but there's those slits that is for breathing purposes, gas exchange purposes for this particular animal. All right, so slits, like I said, will be modified into gills when we're talking about more complex vertebrates. And of course, they function for gas exchange. And of course, gills is only for vertebrates that live in water completely. Because they can't breathe with they don't have any lungs, so they can't breathe in the air. That's why they have to rely on gills. So that's why, with the exception of tetrapods, which are anything that are four-legged and also bipedal, these are things that will vertebrates that will not have gills. So any vertebrates with limbs, we say that they don't have gills, because if they have limbs, that means, and that is also one of the adaptation for living on land. If you have to live on land, you have to breathe air, and gills won't do the job. So this is the gills are, with the exception with tetrapods. Okay, so with tetrapods, what do they do then? So, well, the pharyngeal arches that surround the clefts, which are these little nubs right here, well, then instead of becoming gills, they will develop into parts of the ear, tonsils, and thymus, along with a whole bunch of other structures that we don't need to talk about. Okay, but it's a very complicated situation. We're talking about embryo development, so just simplify it as just parts of the ears, tonsils, and thymus. That's good enough for me. All right, so fourth and last key trait share between all chordates is that there should be a muscular post-anal tail. So this is talking about where there should be skeletal tissues and muscular tissues that form the tail that extends posterior to the anus, coming out of the anus or in the back of the anus, not directly through the anus into the tail. I'm talking about like after your layers of skin and stuff like that. That's where your tail should be. All right, so what is the function of the tail? Well, supposedly they have, I mean, other than humans, that we don't have tails, but for all other vertebrates with tails, they help, for example, those that live in water, they help propel them in water. Without the tail, it's very hard for them to swim forward, and they will be very slow swimmers. The fact that fishes and aquatic marine animals are so adapted to living in water is because they have all these fins, including a tail that enables them to, move in a certain way that propel them very efficiently forward in water. Um, so that's why we as humans who don't have a tail or fins, when we go in the water, we will never beat swimming next to a shark. Okay. Shark will always swim faster than us because that's what their body is adapted for, living in water. Whereas us humans is more so on land. So with our limbs, well, at least we can still swim with our limbs, but just not very efficiently. Okay, so other species that have tails that don't live in aquatic environments, they can use tails for balance and other movements, and some actually for communications and also defense. So if you think about, let's say, monkeys, monkeys are jumping around and swinging around in the trees, so they have to remain balanced with their tail as well, and also use their tail to swing between trees, and sometimes even use the tails to sort of wick off fleas or flies that are flying around them. It's like a fifth limb for them, basically, to help them do some small things that the tail is capable of doing. And for some animals, if you have dogs or cats as pets, we actually can tell their behavior by the movement of their tail. So if their tail is just downwards, not sticking up, that usually means either they're sick or not very happy. I mean, if they're wiggling their tail a lot, that means that dog or cat is very happy. So tails, that's why, can be for communication too, because it's a reflection of their behavior a lot of times. Um, why is it a defense? Well, why tails are defense is because for some animals, tails are actually a disposable part. So, what do you mean by disposable part? Well, it means that if, let's say, a predator catches and bites on the tail, that such as skunks and lizards have the capability, they can actually voluntarily release that tail and then they will escape. Okay, so it's kind of like, you bite on my tail, I'm gonna let you have the tail and I'm gonna run. Okay, so at least their tail is, sort of like a decoy for the predators to keep chewing on, and then their body and basically their whole animal systems are just have already escaped and ran away and saved their lives. So this is why tails can be served as a way of defense. So, for these animals that can voluntarily release their tails, usually they can regenerate their tails later on. So that's nice, which is why they used to say, don't do anything to a lizard because their tails will fly out and they make fly into your ears. I've heard that myth before. Now it won't fly, but they may voluntarily let their tails go because they think you're attacking them. But they won't do that unless they really think you're attacking them. So. Um, Other mammals, such as squirrels and monkey, like I said, will use their tail to grasp, a tree, a branch, whatsoever, and then they maneuver through it. So that's a lot of use for tails that, we humans don't have the benefit of doing. It'll be kind of nice, to have a tail so we can swing in trees, or let the tail go so we can use it as a decoy, something like that. All right, so let's get in and talk about then our specific groups of this phylogenies. We'll start with talking about the two invertebrates we said we'll mention and go into detail about, which is the cephalochordata and the urochordata. So, in common terms, they are called lancelet and tunicate, like I've mentioned before. So, these are actually sister groups to each other. Remember, what do we mean by sister groups? The fact that they are next to each other, that's what we call sister groups, in the phylogenetic tree. So they are sister groups to all other chordates. Oh, excuse me, not like that. Sister group to all other chordates, which is right here. Okay, so these two particular animals are sister groups to all other chordates, and that is your lancelet. Okay, so as larvae, lancelet actually have all four of those key traits we said earlier that is shared by all vertebrates. Even though they're in invertebrates, they have those four traits. They have a notochord, they have a dorsal hollow nerve cord, they have pharyngeal slits, and they also have a postnatal tail. Okay, postanal tail. And but it is after metamorphosis that what they even though they pretty much retains all four of these features even after their metamorphosis, they pretty much still looks the same, but where they live differs. Usually, as larvae, they kind of float up and down and eat planktons. Plankton is their main food source as larvae. And then after metamorphosis, all they will do is they will burrow into and dig into the sea floor and only have its interior end exposed. So it is very rare to capture a lancelet or see a lancelet like that. So when I looked up images of lancelets, a lot of them are actually kind of here's the sea floor or the bottom of the aquarium, and then you have something like that sticking out. Okay, well that's the head of the lancelet right there, and it just stuck itself inside into the substrate like that. And that's usually how you will see them when they're adults. They're kind of sedimentary. They kind of sit there and do their thing, feed on, they are suspension feeders. And then when they will only move when they need to. Let's say a predators come and they need to run, then they will run. Now, it is cilia that will draw water into their mouth. So cilia will draw water into their mouth, and then mucus that forms around the pharyngeal slits will trap the food that's coming in. So that's why they're suspension feeders, because they won't swim around and look for food. They're just gonna sit there and allow this water to bring food into their bodies with their little cilia sweeping them in. And then they will, of course, enter their intestine, which is what that brown part is. That's their intestine. And now gas exchange for lancelet mainly occurs across the body surface. So for this, in this particular animal's case, is not just through the pharyngeal slits. There's some going through there too, but mainly it is across the entire animal's body surface just because they're so thin. Okay, remember we talked about that gas exchange diffusion can happen very efficiently and quickly if the diffusion distance is very short or thin. Okay, so in this case, the lancelet have a very thin body, so diffusion of gases can just come in and out of the entire body versus just a localized area. All right, there's also segmentations within the lancelets and what they call serial arrangement of the muscles within these segmentations. And these are actually formed by the mesoderm, because remember, mesoderm forms muscles. We talked about that back from a previous chapter. So with mesoderm, these particular ones that form these arrangement of muscles are called somites. This is again only for lancelet that they're called somites, and they'll become those segmented muscles. So with those segmented muscles will kind of have this coordinate contractions, which then provides some sort of wiggling movement that allows them to move within the water. But like I said, usually they don't move very much unless they need to as adults. Now, molecular studies have actually shown tunicates, or the Urochordata phylum, are more closely related to other chordates than lancelets. Now, this actually makes good sense is because we think that it was lancelet that came first, and then there's another species, tunicates. Now, these particular tunicates will then eventually, through adaptation or descent with modification, become more complex and eventually into a vertebrate or having a backbone. So that's why it makes sense that tunicates resembles the vertebrates more than they are with lancelets. And molecular studies, remember, is DNA analysis. All right, so the larvae of tunicates, have all the basic structures that we talked about, the four key structures of chordates, and they use their tail muscles and also notochord to swim. So as larvae, they are mobile for these tunicate species. They look like little things like this. Oh, excuse me, the larvals looks like that because their adult form looks completely different. So this mobile larval form will then swim, and they actually have very interesting sensory sensitive cells or sensory cells. One is light sensitive, one is gravity sensitive, and it is through with these two types of sensory cells they're able to search for a suitable substrate out there, and they will attach. Okay, so it's kind of like the sponge or peripheral that we talked about in the last chapter. The larval stage have some sort of flagella, but in this case, they're able to swim themselves because they have a nodal cord now and tail muscles to help them swim. And they look for a specific, suitable spot that they like, and they will attach. And that's the rest of their life is live on this particular substrate. So, which is why after metamorphosis, which is that happens once the larva lancelet, larva tunicate attaches to the substrate. Metamorphosis happen, the tail, the notochord, and the nervous system completely degenerates. They don't need it anymore because they're not moving. A lot of the nervous system, remember we talked about, is for complex movements, fast movement, etc. So because they don't need that, they don't need to swim, tail and notochord gone. And now the organs within the animal actually rotate 90 degrees around. Okay, so completely upside down, where we have um, here's the head of the animal, and then now they kind of goes kind of like this. The head comes down on the substrate. If this is the substrate, the head comes down on the substrate. And that's why the head is right here. And then that's why all the organs is oriented 90 degrees. So that water now flows in through that in-current siphon into the so-called mouth and then out the excurrent siphon. And so in actual reality, this is how tunicates look like upside down in a sense. So, like I said, they will draw that water through the in-current siphon above and then go through all the pharyngeal slits within. And then there's a chamber called atrium for this particular tunicate species. And then the water will exit through the excurrent siphon, through the anus, and then they are able to, with that, water movement, able to feed on whatever comes through. So again, they are filter feeders, suspension feeders that allow things to move through water and trap whatever they can when they come through. Now, even though I said they kind of attach to the substrate once they like it, if let's say there's a predator around, kind of like sea anemones in a sense, back when we talked about invertebrates in the last chapter, they have some sort of escape ability. So they can detach and go somewhere else, and then reattach, I guess, but that's probably a very slow process, and I don't think they wiggle like the sea anemone do. But they do have the capacity to do that. And I already said food is then filtered by some sort of mucus net, and then the cilia will transport that food into the esophagus for it to be digested, absorb nutrients, all that good stuff. All right, so now we're ready to talk about vertebrates. So before we're talking about invertebrates, still the two species or two phyla of invertebrates. So now we're ready to jump into vertebrates. So what are the key traits that are shared by vertebrates then? These are only traits that are present in vertebrates, not all chordates, okay? Because remember, chordates include invertebrates. So those four key traits we were talking about earlier, the nodal cord, hollow neural tube, all that stuff, that was all chordates. We're talking about it's true for all chordates. Now we're talking about key trait that is shared amongst all vertebrates. Okay, so vertebrates have two or more Hox genes. We have already talked about what Hox genes were. Remember, we said that these are regulator of gene expressions, and they tell things where to go, what structure to be formed, and where they should be formed. Kind of, that's why they're regulator genes. And along with transcription factors, now remember transcription factors goes back to protein synthesis, and protein synthesis have transcription and translation. So transcription factors are what's necessary for transcription to even start with protein synthesis. So this is why transcription factors are also very important for gene expression. Gene expression means that you're making a protein from a gene. So in order to start making a protein, you need to have transcription factors binding to what's called a promoter site on your particular gene in order for that gene to be expressed and made into a protein. So those genes that produce transcription factors and signaling molecules were also duplicated due to gene duplication. We kind of talked about that in previous chapter. We're saying that somehow through evolution there are gene duplication that happens, but it's a good thing because it actually gives more diversity, genetic diversity, variability, and also allow for more complex organisms. So if you look here at sea urchins, hemochordates that we talked about, and there's that urochordate earlier we just talked about, and cephalochordates that we just talked about. So that's lancelet and tunicate. See how there's only one row of these rainbow color things? Every row of these rainbow color things represents one set of Hox genes. So these animals all have, these invertebrates right here, all have one set of Hox genes. But when we look at, let's say, a fish, okay, or a tetrapod, look at how complex things get. Okay, so they have two or more sets of Hox genes. That's why you're seeing double or sometimes even triple of those Hox genes within their genome. And that's why things are now can be more complex because of the fact that you have all these gene duplications going on. So, development of a skull, which is very complex. If you've taken anatomy before, the skull is made up of many, many bones and also bone markings. How those are formed is very, probably involves a complex process. And then that's why you need a lot of genes to drive that. In this case, more Hox genes. And then the vertebrate backbone in skeleton as well, because we're moving from invertebrate to vertebrate. So you're all of a sudden developing this backbone within the animal. So, and eventually in higher order of vertebrates, that particular vertebrate bone will actually enclose the spinal cord to protect it. We'll talk about that when we get to later on in the in the chapter. Or if you take anatomy and physiology, you'll see that there's within that vertebrate bone, vertebrate bone right there. There's usually two things sticking out, the processes and then the hole. So within that hole, what we call foramen, is where the spinal cord can fit into the vertebrate bone and allows it to be completely protected by surrounded by bones. So they say that it is these development of a skull vertebrate that actually took over the mechanical roles of nodal cord, because we said when we get to more complex organisms, the nodal cord usually gets reduced or completely taken away. It's because you don't need it anymore. All the mechanical movement the nodal cord provides for landslets, now for complex vertebrates, we can use a central nervous system for that job, the brain and the spinal cord. All right, so third thing, key trait that is, sort of something amongst all vertebrates is the appearance of a neural crest. What is a neural crest? It is a collection of cells along the edges of a closing neural tube of an embryo. What are we talking about here? So look at this picture. In this picture, we're looking back at sort of, I'll draw it instead of going backwards. So remember that we have an embryo with the ectoderm, mesoderm, and endoderm. And so we said that it is the ectoderm that starts moving this way and starts pinching off a little bit like that to form some sort of hollow space right here. Okay, that part of the gastrulation process. So there's that little part, ectoderm, sticking upwards, and you have this hollow space right there. So these cells that are right there within that ectoderm is then known as the neural crest. And if once that neural tube closes off there, completely closes off and form this neural tube, now you have these neural cells that is capable of giving rise or developing, differentiating into a multitude of structures. So these are cells that just, going along the edges of the neural tube in the embryo. They will start to disperse once the neural tube is formed and eventually develop into a whole variety of structures such as the teeth, the bones, cartilage of the skull, neurons, sensory capsules where eyes develop, and also along with other sensory receptors as well that we won't talk about here. But you see how when this is like a baby skull in humans, let's say, um, and that the neural crests eventually become all of the stuff in the baby's skull. So that's a lot of stuff. Again, very, very critical stage of an embryo development where, you have just this ball of cells eventually becoming what shapes like more like an embryo with a head, a tail, limbs, etc. So it's very critical. All right, so now that we got done talking about, some common traits all vertebrates should have, we can go into our first group of species, which are Myxini and Metromyzontida. Okay, so these are just, well, let's go with the common names here. Okay, so common names are hagfishes or lampreys, and they are not very nice looking things because, like I said, as we're going up of these phyla, you will see that the organisms themselves also look more complex. So lancelets was very simple looking. Now we're getting to a little bit more complex, but they still look like a fish of some sort. Um, so these hagfishes or lampreys are known as what's called cytostomes, silostomes. Okay, silostomes are those group that are part of vertebrate species, but they lack jaws. Okay, so cyclostomes is the only group in the entire chordates that are vertebrates. Okay, but they lack jaws. Okay, they don't have a jaw because we did say that that is something that vertebrates have developed is the jaws. So for these group of species, they don't have jaws. They also beginning, the book kind of talks about how they used to think lampreys are not considered as vertebrate or hagfishes are not considered as vertebrate because they only saw a tiny little piece of cartilage. So that little piece of cartilage, they have come to find that that is what's called a rudimentary vertebrae, or what's a small piece of the vertebrae bone that is seen in these particular species. So instead of hard bone matter like the calcium, they are actually then composed of cartilage tissue, which are softer. And also, if they say rudimentary, I would think is very small too in size. But there are molecular analysis, DNA analysis, that indicate hagfishes and lampreys are actually sister groups in the phylogeny. So there they are as sister groups in the phylogeny. For hagfishes, we already said these are species with no jaws. So they have no jaws. They have reduced vertebrate bones, and then they have skull. They do have skull, but just that these things are all made up of cartilage, not very hard, material because they don't need it. They have, for these hagfishes, they look like this. So they have very tiny eyes, also have very small brains. They have ears on that end, and then there's some sort of nasal opening that allows it, that connects it to the pharynx. Remember, pharynx is a fancy term for the back of the throat. So their mouth actually contain what's called tooth-like formations, which means probably they're not very tough tooth, but they look like tooth, and they're made up of protein, keratin. So what is keratin? Keratins are actually proteins that are in your skin as well. So we have keratin in our hair, keratin in our skin, so that we are able to what we call, not we are able to. It gives us waterproof abilities in that sense. Is that that's why we don't need to, I always say we don't need to walk around like a slime man or a frog because of the fact that we have keratinized tissues on top of our skin. So keratin is a protein that allows us to do that. It's in our hairs, on our skin. So particularly, I guess in this species, they use it for their teeth. And then they actually swim in a snake-like fashion by using segmental muscles. They still have those segmental muscles that lancelet have, and the fact that they look like a little, eel, you can imagine them moving like a snake-like fashion. All right, and then the other group is the phyla is then lampreys. Lampreys again have no jaws, they have reduced vertebrae, and their skeleton is made up of cartilage just like hagfishes. No collagen is actually found in their cartilage though. Now, this is something different that lampreys have compared to hagfishes. So usually cartilage have a lot of collagen fibers in them. Cartilage is a type of tissue. They have cells called chondrocytes, which live in little spaces and pockets, and what surrounds these pockets and gives collagen their flexibility, because you can imagine, cartilage their flexibility. Imagine where cartilage is on your face. Okay, so where cartilage is abundant on your face is your nose, and also the tip of your nose, and then your ears. So if you try to move your ears, move your nose, you can move them because they're flexible. And what gives them that flexibility is then the collagen fibers. So technically speaking, this lamprey, they say, don't have any cartilage material in it. So it's probably a very stiff cartilage that we're talking about with these particular animals. They have, though, in addition, they have flexible sheath that covers their notochord. Also, something that is absent for hagfishes. The lampreys have this extra sheath, and they start to have pairs of cartilaginous projections enclosing the nerve cord. So there you go. This one step in species have already showed us there's starting to become more complex. So they say this is sort of like the beginnings of how the backbone was starting to be developed, is because there's that nodal cord, and they say there's a sheath that covers over it, and then there's like projections, cartilage looking projections that's coming out of that sheath, and they say this nerve cord that's underneath then is encased within these projections. Okay, so to me, according to what this describe here, looks like our backbone. So it's kind of like beginnings of our backbone coming being developed. Now, for lampreys, their larva is nice. They are actually suspension feeders. They don't look scary like that. And that's how their mouth looks like. Um, but larvae are just suspension feeders, and some species, though, when they become adults, they are actually parasitic. Okay, they become parasites, and they say when you look it up on the YouTube and Google, they always say that lampreys are invasive species. Invasive species meaning that once you have some of them around in the area, in that particular habitat, they will take over that area if nothing is done to control them. So supposedly, if you throw some of them in the lake or ponds, they will actually start to, reproduce and according to their reproduction seasons, and then they will start taking over all the species and, sort of dominate as species in that particular water body. So what did they feed on? So that's the scary part. Oh, why did the order become like that? I'm sorry. So let me erase some of that stuff somehow. This is a result of lack of sleep, guys. Okay, lack of sleep. All right. So let me take that off. Okay. So I was something I forgot to mention about hagfishes is that they have slime glands. That's something pretty cool about hagfishes, because on the side of their bodies there are these little dots that you're seeing right there called slime glands, and they will secrete a slime when they're feeding on something. That way, that, in a sense, kind of that slimy food that they're feeding on kind of repels other things from trying to feed on the same stuff. More importantly, when they're being attacked, they can actually generate liters of slime in less than a minute, very, very quickly. And what this particular, slime will do in terms of predator, they will actually coat the gills of that predator attacking fish, and then either that fish will retreat, they will kind of swim away, or it can actually suffocate the fish because slime is not very permeable to, water and all that gas and stuff. It's probably another medium that adds to the thickness of the gas exchange surface, which then means that animal is not going to be able to breathe properly and they will suffocate it. So, depending on how big the animal is. And I've seen videos of hagfishes that are attacked by sharks, like really large sharks. Okay, and the moment the shark bites onto this hagfish, the hagfish releases this cloud, humongous cloud of slime, and that shark immediately retreats and then goes away. So, I mean, and you will see in this particular video that I saw on YouTube that any animal that try to come and bite on that particular hagfish, it will just go away. Okay, so it's a very excellent defense mechanism for the hagfish in a marine environment. Okay, so sorry about that. Let's get back to the lampreys. So how do lampreys feed? Lampreys actually feed by clamping their mouth. That's why they have these gazillion rows of teeth, scary teeth that you're seeing there. And they say even their tongue have teeth-like projections on there. And all of that teeth-like projections is kind of like the hooks on your tapeworms we talked about in the last chapter for invertebrates. They will penetrate the skin of whatever prey fish they're swimming around, and once the teeth hooks onto the, let's say, salmon or tuna skin, it will start sucking the blood out of that particular fish. And sometimes it would actually take some tissues along with it too, because if you look at fishes that have been bitten by, or sort of, yeah, bitten by lampreys, you will see scars, like circular scars on these fishes. And because these lampreys are sucking the blood out of that fish, eventually, if there's, let's say, a tuna, even if tuna is a big size, or even a salmon is a big size, but if you have 20 or 20, 30 lampreys all on the same fish, sucking blood out of it, they're pretty much sucking the life out of that fish. And so and that fish, even though it's just getting blood sucked, is not, getting torn apart or anything, they will still die because of the lack of blood. So that's why for lampreys, they are invasive. People don't want them. People are trying to get rid of them, or at least control their population size. There's no way to 100% get rid of them, but they will, at least control their population size by either having these lamprey repellents. They give these to the water bodies, or they set traps for the lampreys to swim in. That way they can get rid of a bunch of lampreys every season, every year, so that they can stop preying on our fishery area. They say they were invasive in the Great Lakes area, and they took out a lot of the fishes there. So a lot of fishes were being killed by these lampreys, which killed the fishery agriculture. Excuse me, fishery department, in which, if you ever see fish prices goes up, it could be because of that. But that was, a while ago when they started seeing this. And hopefully they are controlled now. So they're not very nice, creatures for lampreys. So I wanted to show you that slime that I talked about with the hagfishes right here. That's why I have these two videos. So, those of you that like to play with slimes, or my son like to play with slime sometimes. So they actually produce a big ball of slime when they're being attacked. So this video kind of talks about that or show you that. They don't talk about there's no sound in this particular video, but you see how they kind of swim in that snake-like fashion. And then if someone picks them up and they feel like they are threatened, so there's a bunch of see that cloud starting to come out. That's all that slime. Look at that slime. That's a whole bunch of slime that's just made in less than a minute. Look at that. A humongous ball and film of slime is able to be produced by this hagfish in a very, very short amount of time. How they do that is just they secrete a chemical into the water, and that particular chemical like to absorb water, and once it absorbs water, that's how it becomes hagfish slime. So now here is the scary lampreys, and kind of you get to I wanted to show you how their teeth look like and how they attack fishes in this video.
A snake-like body, sucking mouth with a toothed mouth and more than 100 teeth make the encounter with lampreys unforgettable for humans. This appalling vampire fish is a real dinosaur, as it has existed on Earth for almost 400 million years. In reality, lampreys are not exactly fish, but primitive cyclosami, which occur in freshwater and seawater.
Give me goosebumps.
They are nothing but predators with a length of more than a meter and the weight up to 3 kilos. During a year, one fish of this species can consume up to 20 kilos of fish, whereas their life expectancy is 5 to 7 years. Actually, lampreys are parasites. They cling to you and through the scale penetrate into the body of their prey, inject digestive juices and anticoagulants which block blood clotting.
Kind of like leeches.
Therefore, fish which are still alive start to get digested, and lampreys only need to suck into this cocktail of guts.
Okay, and that's pretty much it for that video. Just something pretty short to show you how it clings on to the fish like that, and it straps on there and leaves a scar there for the fish. So that's just one lamprey on one fish. Imagine like 3, 4, 5 of them on there sucking blood, out of this fish. That fish is gonna die pretty quickly. So
a snake-like body.
That's why, they're not very welcome for those lampreys. All right, so we're done talking about the landslide, tunicates, the hagfishes, and the lampreys. So now we're on to, very interesting, sharks. Oh, and I wanted to talk about the fact that hagfish slime actually has been investigated and researched by engineers where they want to develop some sort of space filling gel that can be used to stop bleeding during surgery, which I thought that was pretty cool because we talked about how sponge create these antibiotic and things that are anti-cancer treatments. So somehow hagfish slime have also similar capacity to do something like that, something beneficial for the human race. But I don't think lampreys have anything beneficial so far. But they are both hagfishes and lampreys are the sole survivors, from the Paleozoic era. Remember, Paleozoic era, or somewhere around there, is about 400 million years ago. These species have thrived for quite a bit, been able to survive through mass extinctions that we talked about. So they're pretty powerful creatures, even though they're not that complex. All right, so on to the next group of species that we'll talk about, or chordates that we'll talk about. All right, so Nagthosome. Nagthostomes are actually vertebrates that now have jaws, because when we talked about lampreys and hagfishes, they did not have jaws. But vertebrates, remember that they do have a key trait that's developed, which are jaws. So nagthostomes means they are vertebrates with jaws. And this particular group here, or phylogenetic group here, it includes sharks, ray fin fishes, lobe fin fishes, amphibians, reptiles, birds, and mammals. So this is one of those longer ones when we're talking about the length of the groups. So here is lengthosomes, and lengthosomes includes this whole group of phylogenetic taxon. So that's why it includes all those animals. All right. So remember, jaw is something that is now appearing in these species. So what are jaws? They are hinge structures that allow the animal to grip the food firmly, because without jaws, that's why that lamprey have to have all these rows of teeth, scary looking teeth. And that's because that's the only way it was able to grip very tightly onto their prey. But when you have a jaw, kind of like imagine a pincer or a clip, that's how you can clamp down on whatever prey you're trying to eat. And then with teeth on those jaws, you can actually be able then to slice and tear pieces of tissues or flesh off of the prey. So that's why jaws are a very important advancement in the vertebrates when we're talking about how they're able to survive and become very good predators. All right, so common ancestors of all nactosomes underwent another round of duplication of Hox genes. So that's why nactosomes, a lot of them have at least four sets of Hox genes that allow them to have all these more complex things that start to come into play when we talk about these species. And then they say that their entire genomes seem to have duplicated as well. Remember, genome means that it represents all the DNA and chromosomes within that species. So because of this complexity in their genes now, with gene duplication and the entire genome being duplicated, so it's not just the Hox genes, but also the entire genome, this is probably, they think, what enabled the origin of jaws. And although all the other novel features and structures we'll talk about that vertebrate animals are able to do from nagtostomes onwards. So the forebrain of nathostomes are also enlarged compared to other vertebrates, which means they can do more nervous system things, which has to do with sensory or motor. So in this case, there's a lot of enhanced senses of smell and vision. Remember, with invertebrates, we talked about animals with simple eyes or just one lens or things like that. But now there are animals with very excellent acute vision, and also what allows for some, them being good predators. And then there's also very heightened smell as well. We always talk about smell of a dog. So smell of a dog is much more enhanced compared to invertebrates that we've talked about. And they are able to smell so many different chemicals, all because of gene duplications. In at least in the sense of evolution. And then for nactostomes, in terms of aquatic animals, they have developed something called the lateral line system. What is lateral line system? This is kind of drawn on this fish right here. Lateral means on the side of the body of the fish, and it's a system and network where there's these organs that form a row along each side of the body, and they are sensitive to vibrations in the water. So even though, fishes may not be able to hear, okay, especially sharks. Sharks can't hear, they don't have our eardrums like us for vibration through air, okay? But what they can feel is vibration of water, okay? They can't feel vibration of sound going through air, but they can feel vibrations moving through water because of this lateral line system. And if you look here, they actually have sensory organs that look very similar to humans when we're talking about our inner ears that senses movement. We talked about in anatomy, we talk about an area within the ears called the semicircular canals, which then inside the semicircular canal, there's a structure called ampulla. Within the ampulla, there's these ampullary cupola that allows and feels the movements or spinning of your head in rotational movements. So in this case, there's that cupola again, but in this case is then for sensing vibrations for the fishes in water. So that's pretty interesting. Something that is probably, common amongst between humans and vertebrates. And we will probably find more and more traits that we share because now we're getting closer and closer to the complex Homo sapiens that we are today. All right. And then for, again, aquatic animals, now there are paired fins and tail as well, along with dorsal, ventral, and anal fins. I'll talk about what all those are when we get to the sharks, but just think of dorsal as the top, ventral as the tummy side. So there's a back, a fin on top of the back. There's also tummy fins, and there's also anal fins, okay, that is around at the back or posterior parts of the animal. And now they are stiffened by bony structures called fin rays, because they used to be just soft flappy things, but now there's bony structures within the rays that start to make it harder and more resilient. And this is what allows for faster swimming. Okay, imagine, I mean, this is why when we want to swim fast or dive, people put on flippers, because those are allows for more movement and wave-like movement that allows you to go deep into the ocean and swim a little bit more than your tiny little feet with not a lot of surface area. So having fin-like things that allows for faster swimming. But if fishes have floppy fins, that's not going to help them. Go sort of in that floppy motion very quickly. So, because they gotta sort of swing their fins. This is where I'm like, I wish you can look at me, but I'm waving my hands so it looks like a fin. Yeah, that's how they swim through the water. But when you're talking about floppy fins, then they won't be able to move water very efficiently, and they can't swim as fast. So this is a very good improvement for faster swimming of fishes compared with, let's say, aquatic worms that we've been talking about. Um, and then there's also thrusting and steering control now. With all those fins everywhere, top, bottom, left, right, back, now you have a lot of control and also balance when they're trying to chase a prey or escape from predators, because now it's kind of like a plane. Now you have fins everywhere to help you know exactly where to go, how much to turn in a fast swimming motion. Now, by about 420 million years ago, Nagnosomes then have diverged into three lineages of jaw vertebrates. And they're the conrychthians, the ray fin fishes, and the lobe fins. Now, it is 1 hour, 6 minutes. I'm going to stop here before we move on with the conrychthians.